Okay, what was I going to say about this? Okay. Okay. I'll make something up. No, I'm just kidding. Okay. So one of the things that we're going to be looking at is what happens when substitutions or mutations are made in DNA sequence. And what we're eventually going to try to get to is what effect does that have on presumably the amino acid produced, the protein, and potentially phenotype, and then fitness. And there are lots of steps from that mutation in the DNA sequence to the effect on the phenotype and then to fitness. So this is, again, not a simple thing, but a lot of times when you think about the 4 base pairs of DNA, right, you have adenine and guanine, which are called purines. Again, I'm not going to have an exam question. The purines in DNA sequence are blah, blah, blah. I'm not going to ask you those sorts of questions. But these are the purines, A and G. Cytosine and thymine are pyrimidines. And again, I'm not going to ask you what those all mean, how that's defined. But a lot of times, and you'll find this, is that substitutions that go, a mutation that occurs between adenine and guanine, right, a change in a nucleotide from adenine to guanine is called a transitional substitution or transitional mutation. And it can go in both directions, A to G, G to A. That's why these arrows go back together. Transitions can go, obviously. Transitions in the pyrimidines can go in either direction. The other thing you can get are what are called transversions. And transversions are defined pretty easily, right, by why is it called a transversion? Because it's a change from a pyrimidine into a purine or vice versa, okay? And I'm not going to go into all the details, and I'm not sure I even remember why, and you guys probably, have you had cell and molecular? So tell me why. Which one of these do you think is more common? And in DNA sequence data, which do you think is more common and which is less common? Okay. Well, it turns out that when you look at ratios and substitution ratios, transition ratios tend to be much higher and occur at a much higher rate than transversion ratios. And there are lots of reasons why. Yeah. So these are not as common. And transitions tend to be more common. Okay. So again, what we're looking at is basically mutations, right? Changing from an A to a G, G to A. But transversions do occur. Depends on what kinds of organisms, but they occur at a much lower rate, and there are kind of structural reasons why. I forgot in. I realized as I was going through this. So what we're going to sort of look at is we're going to be looking at mutations and what effects they might have and whether they're beneficial, deleterious, or just neutral. And these are convergent evolution in 2 high-flying geese. It turns out, and I didn't look at the original paper, but this is in your book. It turns out, and we'll be defining what a non-synonymous mutation is, but it turns out in both these Himalayan, these are different species, Himalayan goose and this South American Andean goose, they have a non-synonymous mutation in a particular gene that's involved in oxygen binding and increases the sort of ability to attach oxygen and make oxygen available in tissues and all those sorts of things. And they have this mutation that really gives them much improved oxygen binding and obviously leads to higher performance at high altitudes when oxygen can kind of be an issue. So it turns out these 2 things, and we'll be explaining what a non-synonymous mutation is, but this would be an example of a mutation that is bad, neutral, or beneficial. Beneficial, okay? So this is going to be an example. And we're going to be looking at some data that looks at, are most mutations kind of neutral? Are they generally deleterious? Are they in general? And it depends on the organism that you look at. But this is kind of a cool example of, and I think this is basically base pair changes in this gene of about 3 to 4 base pairs, and they were all non-synonymous that resulted in this really change in this oxygen. I think it was an oxygen receptor. Okay, so let's talk about how we think about the effects of mutations and what we mean by synonymous and non-synonymous mutations. So Here again, we're just going to primarily be focusing on, here's DNA. We obviously have a double strand. We're only looking at 3 nucleotides, and we're only looking at a single codon. So here's the sequence complementary, TCA. The other strand is AGT, complementary. So here's an example where you go through transcription and translation. There's no mutations, right? And you get a serine being produced. This is the case. This is kind of this is where you consider this the ancestral state, where there are no mutations. Here's a synonymous mutation. This is what we call a synonymous mutation. That is, where did the mutation occur? At the 3rd base pair, right? Okay. So there was a substitution, let's just say for a C, complementary, we give it a G, and then you then produce UCG when you translate that, and what kind of codon do you get? It's in the same position. What do you get? And you can go back to that circle, and you can find that TCG and TCA code for the exact same kind of amino acid, serine. So the new codon encodes the same amino acid, so there's no change in the amino acid. What would be your prediction about the effect of a synonymous mutation on the fitness or reproductive rates? Do you think it'd be deleterious, beneficial, or no effect? No effect. This is the case. These are often what we call neutral mutations. Most synonymous mutations, especially at the 3rd base pair of a codon, are synonymous and have no effect on phenotype, on anything. Okay. Here's an example of what we showed previously, what we said occurred in those 2 geese, right? A non-synonymous mutation. And you'll notice it's not in the 3rd base pair. Where is it? It's a mutation that occurs in the 2nd base pair of a codon. And here the messenger RNA switches from a U, UA, and what do you get if you go and look up that code? You get a new amino acid in that polypeptide chain. All the others, assuming that's, so there's a change in the amino acid at that 3rd amino acid in. The new codon encodes a different amino acid, and there's a change in the amino acid sequence. So, again, it's harder to predict what the effect is, but it might be that probably it's going to be either beneficial, or you think it's neutral, probably not, or it could be negative or deleterious. Here's a real obvious one. So here you have, this is from TTA, you go, no, they're all different. So you have a mutation here at the 2nd base pair that actually produces what's called a stop codon. That's, if you go back to that pi, UAA codes for what's called a stop codon. What do you think happens to the protein? How do you think that protein's going to do? That basically truncates the protein, right, stops it, and you're basically then left with 2 amino acids. Do you think the effect of that is going to be pretty dramatic? The effect of that is going to be extremely dramatic, usually very negative. Okay? So stop codons in general in functional proteins are not good. I'm not going to talk about that. Ignore that one. We're going to be talking more about this, but one of the things we're going to be talking about is how crossing over and recombination between sister chromatids can produce sort of, it's not new genetic variability, but it rearranges the alleles and the combinations of alleles. So it's kind of an important source of genetic variation, and we'll be talking a little bit about this. But when you think about crossing over and recombination, so let's imagine we're just looking at, here are the homologous chromosomes and sister chromatids and all those things, and don't worry about how each one of those defines. But so here you have 2 sister chromatids. And what's this thing right here represent? And do you even remember what phase of meiosis this occurs in? You've all forgotten that? I forgot it within an hour after I took my exam. I can't remember. Was it metaphase? Does anybody remember? See, this is why This obsession with memorization is not good. Okay. I shouldn't be saying this stuff. Okay. The important point here is not, when this occurs, but the process and the outcome, right? And this is when the sister chromatids, right, synapse, and they exchange arms of these chromosomes. And what you'll notice is when there's the duplication event where you have these homologous chromosomes, then they get duplicated, producing, these daughter chromatids of each, right? And there's nothing different there. But actually what happens is when these two sister chromatids recombine, right, what they do is they're exchanging these arms, right, through that crossing over event in a certain phase of meiosis. Yeah. Prophase 1, okay, good. Remember that. That's going to be on the exam. Those are the sorts of questions. Yeah, I'll say, did you know that or you had to look it up? Okay, okay. So again, and obviously what's important to me is the outcome, right? What you've created here is a combination. Each one of these represents a locus or a gene, right? So here's the A locus, the B locus, and then here's the C locus way out on the distal end of these chromosomes, right? What's important here to me is that what that crossing over event does is, has it created new combinations of genes on these sort of sister chromatids? Yeah. So instead of small a, B, and C, now you've got A and small b now in combination with big C, and just big A, big B here in combination with small c. That's different than what you saw before the recombination event. So recombination can kind of rearrange and put different allelic combinations together. And it's an important thing, especially when we get to other parts of the course. Okay. Let's just talk a little bit about the effects of mutations. And I'm just going to go through a couple examples. And they really kind of illustrate a basic take-home message. And this is study based, I think it's on a bacteriophage, okay? And blue represents what are called replication genes, green are maturation. So these are viruses and things like that that have to go through host entry. They then, mature. They have to produce what's called a capsid to get, and then they have sort of other genes that are involved in breaking the host cell and extrusion and then transmission. So these are a lot, and virus gene, these things are fairly easy to sequence. They're not very big, so they're easy to kind of characterize. And basically what they did is in this particular example, they kind of created spot mutations through various mechanisms and then looked at the effects of these mutations on the fitness of descendant populations of the virus. So they had ancestral population that had none of these mutations, and then they compared their relative fitness. Okay. And it's not too important. All along here, the third base pair mutations, these are second base pair mutations, and these are first base pair mutations in these various genes that are associated with replication, maturation, capsid production, and then extrusion. And so you can kind of see the mutations are scattered throughout the genome in various parts of the first, second, or base pair and these various things. The key take-home message here is pretty simple. What's the take-home message? If you had to characterize this graph, what's the most common outcome of a mutation? Kind of neutral, okay? That is, what this means, fitness of 1, what do you think that means in terms of those, I hate to say daughter viruses, but I guess I will. The daughter viruses, if their fitness is 1, what does that mean? What's their reproductive rates or relative to their ancestral state? It's exactly the same. Okay, so that would be the case where there's no difference in the reproductive rates of that virus compared to their ancestral population. But actually, if you count up all these things, what would you say? In fact, most mutations tend to be kind of negative. Okay? Well, I don't know, it might be pretty close. Anyway, so what you can see is there are a lot of mutations down here, and what this means, anything below 1, what does that mean about the daughter DNA in terms of her fitness? It means it's less than the ancestral population. So what this suggests is some mutations are deleterious. That's their negative. They reduce fitness by about half. That's a pretty strong effect. So a lot of them were deleterious but not lethal, but then you had over 20% of them were lethal. And guess which ones those were? Those were primarily the ones that occurred in the first or second base pair, and especially ones that produce stop codons. Those were absolutely lethal. And there were some other things. So what does it look like in terms of the fraction of mutations that are beneficial? Not so much, right? Okay, so that's the rarest class, at least in DNA viruses in this particular study. So, and this is kind of a general result from a lot of multiples and hundreds of studies. Most mutations are fairly neutral or deleterious. This is kind of an interesting study. This is from a heat shock protein. I think it's from humans. It's in your book. I don't remember. But heat shock proteins are, I don't think it is humans. It can't be humans. But heat shock proteins are very important, and they're really important in sort of high temperature environments in terms of buffering against lots of negative effects of high temperatures. And so I'm not going to go into all the details, but they did this study. It must have been a bacteria or something like that. One study done at 36 degrees Celsius, one at 25 degrees Celsius. And here are the number of mutations along here. And what's represented in blue are those that had synonymous, the frequency of synonymous mutations, what are called nonsense mutations, which are basically the stop codons, okay? And then all mutations. Okay. What do you know? Let's just focus on a couple things. One is most mutations fall to the left of one, right? So in general, most mutations in general are pretty deleterious, Are some mutations more deleterious in one environment than the other? I probably should never say this because now you're going to get confused, but where are mutations more deleterious? In which environment? And how do you know that? So you kind of have to look at this tail, right? And then how many individuals are below this number one, right? What you'll notice here, and the extent of it down here. So what you'll notice here, up in the kind of not really high temperature environment, most of the mutations are deleterious, but not as dramatic as in the warmer temperature. Do you see that? And especially what you can see, there are a bunch of nonsense mutations that have really strong effects on fitness. See, all the reds are right out in here, and the reds are over down here where they reduce fitness a lot. But what you'll see is this tail is much further out here where you have really strong effects on fitness. Okay, so why is that? It's fairly straightforward. It turns out that these heat shock proteins are really important, right, in dealing with high thermal environments. Turns out their ability to work is diminished, or the mutations have a much greater effect on the ability of that heat shock protein to work in those high temperatures. So it was kind of, and this is in your book, it's kind of an interesting study that showed, obviously most mutations are deleterious, but the effects are much more dramatic in kind of a more extreme environment or a hotter environment. And it basically is that heat shock protein doesn't work as well, and those mutations affect it a lot more in that kind of environment. Okay, so those are the discussion questions, blah, blah, blah. Okay. Let's spend the rest of the time introducing you to dihybrid inheritance. Do you know what that is? When Mendel not just crossed individuals with one trait, but with two. Did you do those probably did those in freshman biology, right? So we're going to kind of go, we're not going to spend a whole lot of time on, I'm going to give you a homework, I'll give it to you on Tuesday. There are going to be problems on it that, I haven't gotten to the lecture material, so it'll be due at a later date, but you can start working on it. And it'll be homework that you turn in and be graded on. And there'll be similar sorts of questions on the exam, so hopefully this will be helpful. Okay, so let's. myself comfortable? Okay. You don't need to see me, right? Okay. You don't want to see me. Okay. Okay. So let's imagine we're looking at something. We're looking at, let's go to corn, the dreaded corn. Okay. So we're going to be looking at sort of seed color as one phenotype, and then seed shape. So those are the two phenotypes. So we're envisioning we're going to make it fairly simple. The seed colors are going to be potentially phenotypes that are going to be yellow. You actually are doing this in your, I probably got it wrong, I don't care. Yellow and purple, and seed shape is going to be round and wrinkled. So we envision these are the two potential phenotypes that we observe in corn in this particular cross, and we'll show you an example. So let's kind of just present this as here are the, and the easiest way to do this is what are called, oh geez, I'm blanking, on a test cross, where the two parents, one of them is homozygous recessive at both genes. It makes it a lot simpler to do, and the outcomes are real simple. You can do Punnett squares with mating two individuals that are double heterozygotes, and you'll have a 4x4 table, and then you got to translate all those genotypes into phenotypes, assuming dominance. So what we're going to assume here is, what did I decide? Okay, yellow is dominant in this case, the gene coding for that, and round is dominant. So this is the assumption, and we'll show you the genotypes. Okay, so let's imagine you have one male parent and a female parent. We are going to assume at this point that the genes coding for seed color and seed shape are on different chromosomes. That is, they are not linked, and they assort independently. Okay? So let's say the male is, we're going to say it's heterozygous. And the female is going to be completely homozygous dominant, homozygous recessive, okay? She has lowercase of each. Okay, so what we're assuming here is these two traits are encoded on separate chromosomes, and that big Y is completely dominant to small y, big R is completely dominant to small r, okay? So what we're going to do is we're going to put there big Y, small y, okay? And the other combination of genes over there. And that is homozygous recessive for both genes, for both traits. Okay? You all want to do a Punnett square, don't you? I'll show you an alternative. And what I always did, I just memorized what the outcome was, and that's a really bad strategy. Remember the outcome depending on the kind of cross, but don't do that. Okay, so let's just look at this one. When you think about this individual right here, do you really need to put 4 kinds of gametes in a 4x4 Punnett square for that kind of individual? What kind of gamete is that individual going to produce? Yeah, under a reduction division, right? What you're going to get is 100% of what? Small y, small r in the haploid gamete, right? This one's a little more complicated, right? What are you going to get there? And the way to think about it is how meiosis works and the expectations under normal meiosis and things like that. But what do you think you expect from normal meiosis under typical Mendelian inheritance? what kind of things are you going to, what kind of gametes, haploid gametes, are you going to get out of that individual? Yeah, you're going to get one of each, and you're going to basically get 4 kinds of gametes, aren't you? So the way to do this is pretty simple. Just kind of boop, boop. So you're going to get 1/4 are going to be what? Big Y, big R. Take that big Y, right? Times that one, you're going to get 1/4 big Y, small r. And again, watch the Khan Academy video, see how all this is done. And this is assuming all sorts of normal Mendelian meiosis, and if these are located on separate chromosomes. You're going to get 1/4 small y big R and 1/4 small y small r. Okay? So so how many haploid gametes are you getting out of that parent? 4, right? Now what do you want to do? You want to know what your expectations are in terms of offspring from that cross, right? Okay. How do you want to do that? You can do a Punnett square if you want. How do you want to think about this? Here are male gametes, right? How many female gametes do you get, and which ones can they fuse with? Only one kind. So how many genotypes are going to be produced from this cross, assuming no mutations and that the genes are located on separate chromosomes? 1/4 of each, right? Okay? So let's think about that. So you're going to expect 1/4 to be big Y, take that one, right? And then where do you get a small y, right? Here's the big R, fusing, then you get, okay? So that's 1. Then you keep going, big Y, small r, small r. Then you go down here. You got small y, big R, small r. And then the last one, what are you going to get from that one? Small y, r, small r. So those are the 4 possible genotypes that you get from that cross. What are the phenotypes of each one of those? That's going to be 1/4 expectation from that cross in terms of the phenotype of the offspring. That's the genotype. The phenotype would be yellow round, okay? Remember, big Y, the big ones are dominant. What kind of phenotype is that? Yellow wrinkled. Here are purple round. And the last one, purple wrinkled. Okay? So the expectation from a cross between a double heterozygote and a completely homozygous individual is, a 1-to-1 to 1-to-1 ratio of each particular genotype and phenotype. Here's what I want you to think about. Imagine you did this cross and you got 1,000 seeds. Here's what you observed of, oh, geez, now I can't even remember. Yellow, round, yellow, wrinkled, purple, round, and purple, wrinkled. And when Mendel did this experiment, here's what he got. He got 499, no wait, yeah, I can do that. 499 of those. Well, I should have done this beforehand, but okay, if I make a mistake, who cares, right? The outcome should be pretty straightforward. He got one of these, 499 of those, and one of those. Let me just ask a general question. Does this fit the expectations on the from the previous thing you did? What are the expected numbers of these, assuming this, 1/4 of each, right? The expectation will be 250 out of 1,000, right? 250, 250, and 250. So clearly something went wrong here. And it didn't fit the expectations of, for example, Mendel. And so what we'll talk about next time is what can result in that. And do you guys know what that is? What assumption was violated? What did I presented previously? So little hint, I had the genes located on separate chromosomes. We'll talk about what happens when they're located on the same chromosome. And we'll go through an example of that. That's what's called linkage, and we'll talk about that next. Okay. So there'll be another PowerPoint, but we're going to start getting into Mendelian genetics and population genetics pretty heavily next. Okay.